Hello, everybody. Welcome to this week's webinar on FungiDB and the resources and tools that we have to offer. I will go ahead and share my screen, so let me know if you can see it. If somebody could, let's see, here we go. So you should be able to see my screen, and we will start just in a few minutes to let people join the webinar, as we have quite a few registered. Meanwhile, we would like to run a poll. So normally we start our webinars with live surveys, and today we would like to ask you about which organisms do you work with. So what's shown is a live survey link on the right, as well as QR code, and I'm also posting this in the chat right now. If you click on the link, please go ahead and answer while we are waiting on others to join, and we could actually see it in real time to see which model organisms are worked on today's call. I can tell that people are still joining, so welcome for those that are still joining us. Go ahead and fill in the live survey, the link and the QR code on the right. And while we're waiting on others to join, I would like to just say a few things about housekeeping. We have quite a few people registered today, so most likely we won't be able to answer if you raise a hand, but we do have several members of the outreach team on call today joining the webinar, so that if you have a question or comment, please go ahead and type it in the questions panel, which is within the GoToWebinar control panel. So on today's call, we have Omar Harp, Director of the Outreach in Education for ViewPathDB. We also have Gloria Gerardo Calderon, Sam Rund, Suzanne Warrenfeld, who are outreach specialists and will be answering your questions in the background. Just a quick note that today's session is being recorded. So if you have to leave early, you can always come back and listen to it later. We will post a link to our YouTube channel, and it will also be available from the FungiDB webinars page, which can be located under the Help Learn How to Use ViewPathDB site. So you notice we have a list of previous webinars, and if you navigate to one of them, most of them will have a link to the YouTube channel, and also if there are any additional link outs, such as handouts, they will be listed here as well. So let me go ahead and bring up the direct poll, which is going very nicely. Thank you very much for filling it in. And lastly, I guess, if you're interested in receiving a certificate for today's attendance, we will be sending out an email with certificate in a day or two. So if you don't get an email by Monday, let us know, and we will help you out. All right, how are we going? So I have about 140 people, so we have a good number. So let's go ahead and start. So my name is Evelina Basenko. I am a FungiDB outreach specialist, and today I'm also joined by several PIs of the ViewPathDB project, including FungiDB. And these are the PIs are Mark Caddick, located at the University of Liverpool, and also David Roos at the University of Pennsylvania, that would like to say a few words before we delve into the demos. David?
Hi all. Thanks, Eve. My name is David Roos, and I'm one of the PIs associated with this project, as Eve indicated, and I will keep this to a very few words. The Bioinformatics Resource Center program is supported by a variety of sources, including a contract from the National Institute of Allergy and Infectious Diseases at the NIH, but also with parallel support that has been particularly important for fungi from the Wellcome Trust. And because of the way the BRC program in the US has been structured, we have recently restructured to make fewer, larger programs, including support for a variety of different organisms. This has led to some infrastructural changes that gives rise to the new website that you have probably seen before for FungiDB, and we'll certainly spend more time talking about today with Eve and others from the team. This merger has been a behind-the-scenes effort that has consumed most of the last 6 to 8 months, and we're really glad that that's finally complete and allows us to turn our attention back to the needs of end users. The infrastructure is not something that I want to go into in any detail, but I will point out that it brings many benefits to the project. For example, for the fungal user community, ensuring that FungiDB and Ensemble are entirely consistent, as all data is processed through the Ensemble pipeline. And we expect that that will improve our ability to process and load genomes and functional genomics data more rapidly and consistently in future, hopefully incorporating a wider range of the genomes that have been sequenced by JGI over the years as well. So that's one concrete example of benefit to this underlying infrastructure, and we look forward to your feedback. Mark, did you have anything that you wanted to add from the standpoint of fungal users?
Yeah, a couple of things really. The FungiDB is there for you, and it's not there for us. So we're here to give you what you want. So please communicate with us, share what you require. If there's new initiatives, we want to know about them. So Eve, who's running this call, is the outreach person who works mainly on fungi, so she's the person to talk to. If you want sort of different facilities on the website, talk to us and we'll see if we can help you with that. If you want to analyze your own data, you can do that already. I'm sure Eve will talk to you about that. But it's really important that you communicate with us so that we do the best job for you. So we've got outreach groups which will be restarting quite soon. So we'll talk to various people in the community. If you want to be part of those, then please let us know and we'll include you in that. And if there's important data or even small information about specific genes, let us know. We can then make that difference to help everybody get the best out of the website. That's all I've got to say, really.
That sounds good. Just to actually follow on from what Mark said, I know the fastest and best and easiest way to contact us is to use the Contact Us link that's up at the upper right-hand corner on any FungiDB page. You're certainly welcome to email Eve directly, or Mark or myself, but the most reliable way to get a response is to click on the Contact Us link. That's your one-stop shop for how do I find this about a gene? How do I download all this stuff? I have data that I think I might want to load. Would you be available to run a Zoom or GoToMeeting call with my lab meeting, whatever. So I think with that, we can probably mute ourselves and turn things back over to you, Eve. Thanks very much.
All right, thanks very much, David and Mark. All right, thanks very much to those that have contributed to this survey. If you're just joining, welcome. There's a few links in the chat box that will take you to the questions displayed here. All right, so let's go ahead and move on. And we will start first with a brief introduction into FungiDB landing page and its components. I am on the FungiDB beta site. This is our new site that we released in the release 47. And the web page has banner that contains logo, and this is also clickable banner. So if you navigate away from the main page and want to come back, all you need to do is just click back on the banner. The new functionality is right here shown at the top, which is a site search. You can use gene IDs or text term to search various records and terms in FungiDB, and I will demo it in a little bit. We also have a list of menus underneath this site search, including my strategies, specialized searches. For example, these searches at the top are also the same searches that are listed right here on the left. And these are categories of searches that include pre-computed and results generated by in-house workflows, such as SNP analysis, RNA-seq evidence analysis, etc. And we will take a little close look in a few minutes. We also have a menu for tools that allow you to quickly navigate, let's say, to Genome Browser, JBrowse. You can further explore and analyze your data using My Workspace. Notice that if you navigate to Galaxy, you'll be able to analyze your own data in the Galaxy instance, and this is private workspace, so no one sees or has access to your data but you. And what is really cool that once you analyze your result, once you analyze your data, for example, RNA-seq and Galaxy, you can bring it back to FungiDB and integrate it into My Datasets section. Again, this is a private workspace for you. And within this workspace, you can essentially create searches that we offer within FungiDB to further mine your data. We also have menu for the datasets where you can learn about different types of data or download files. In the section About Us, we provide a little bit more information about the project, including how to submit data to us. Quite frequently, we are asked questions about, well, how do you learn about new data sets out there? How do you prioritize them for loading? And the short answer is that while we try to survey the current literature and identify new cool data sets, this primarily is feedback from you, from the emails that we received when people go to the Contact Us form, from communications during conferences and help booths, personal communications. So it's essentially feedback from you. When you let us know which data sets should be prioritized for community, we track these data sets and schedule them for integration. This section actually has two different subcategories. For example, this section explains how to submit data to us, but also walks you through about the types of data that we currently have. And if you don't have if you don't see a data set that we currently support, let us know by contacting us, and we will do our best to try to develop new features and tools. And there's also more information of how you can alert us about different types of data. And to read more about the process and our release policy, click on the second link, which is redirecting you to the PDF document. We encourage you to let us know about datasets well in advance of their publication. And we never release data essentially without your consent, as all new data being loaded to a password-protected QA site. And while it undergoes QA, we also work with you to make sure that you're happy with how dataset is being represented before it sees public release. All right, we also have help section, and as I have shown you before, you can navigate to learn how to use ViaPathDB to access a list of webinars, either past or future webinars. We also have sections that are dedicated to provide you with learning materials, such as materials from workshops or quick tutorials to get you started. Also methods about how we process data in FungiDB. And if you would like to invite us for a short lab meeting or a mini workshop, let us know and we will get back to you as soon as possible. Also within the banner, we have a section that links to the social media, such as Twitter, Facebook, and YouTube. And there's a section where you can log in or sign up for an account in FungiDB. The account is not required to be able to use the resources offered in FungiDB except when you're working within the Galaxy. However, having an account, which is free, helps quite a bit, because if you would like to create a search strategy and then share it with someone else, such as a colleague, or show it to other people what you have done, you can create a unique URL to share, and that will be stored in your account. All right, so this is a sort of short overview of the banner. Next, we have specialized searches on the left, which is you can explore by essentially expanding all sections. And if you're interested, let's say, in deploying a search for a signal peptide, you can also use filter to identify and quickly go to this particular search. In addition, we have a central panel with overview of resources and tools that houses essentially a collection of cards. There are mini tutorials and designed to get you started with learning how to use FungiDB and create, for example, complex search strategy as shown here. In addition, we have resources at the bottom of the screen, and these are a little bit sort of expanded versions of tutorials, and we are working to create more tutorials and exercises for fungal organisms right now. In addition, we have a news and tweet section, and this is our way to communicate about the types of data sets that are being released. And notice at the bottom of the main page, we also have a community chat section. We are still playing around with this, but the goal is essentially to enable the community chatting with each other. So if you have a question and somebody in the community knows the answer, hopefully they will answer or share additional information with you. And one more thing that I wanted to mention, just to follow up what David and Mark have said, we are going to resume the community forums quite soon, so stay tuned for future communications. We welcome everyone to join these conversations and let us know about cool new high priority data sets for the community, or even various tools that we should implement in FungiDB. Okay, so are there any questions so far before I move on to demoing a few searches in FungiDB? Omar?
Sorry, I was just addressing some questions about, there are a couple of people who had audio issues, so that's fine. I think those are local from their end because the audio is working for most people. I've checked on that. And also, there weren't specific questions about the database yet. There was a comment from Megan Jankowski saying that she can vouch for your comment about us being very respectful about uploading and making sure we coordinate with data providers with the publication or pre-publication as well. And so that's great. Thank you for that comment, Megan.
Oh, fantastic. Thank you very much. Okay, so let's go ahead and I'll walk you through a few searches in FungiDB. So notice that I mentioned before we have a new functionality that's called site search. And you can search either for a specific gene ID or for a term. Let's go ahead and create a search for ergosterol and click enter. So notice the page now refreshed, and you have essentially a list of results. And these are records or terms that are found in FungiDB that include a search term, ergosterol. See what happens to this button once we navigate through the categories within the filter results. Right now, this button is shaded to where you cannot create a search strategy, and that's simply because you have a number of results on the left. And before you can create a search strategy, you need to select what is it that you would like to concentrate your your attention on. So let's say I would like to identify all genes that have ergosterol mentioned somewhere in the product descriptions, and I'll click on the apply to select that filter. And you know what, since we have quite a few genes, I'll go ahead and limit my search to a specific genome for Aspergillus fumigatus, AF293. And let's click apply. Here we go. So now we have identified 11 genes that fit this criteria. And now by clicking on the export this as a search strategy, I can essentially create my first step of in silico experiment. And this is quite simple. And from this point on, you can click on add step and expand your strategy by using any other types of data that are integrated in FungiDB. So that's a quick example of how site search can be used to access and filter through your results. In addition to site search, we also have dedicated or specialized searches. And this is somewhat different from site search. For example, you can navigate our records for SNPs, such as compare SNPs within a group of isolates, or you can even look for genes within any of these categories, such as transcriptomics. So let's say we are interested in looking at the RNA-seq evidence for Aspergillus fumigatus. So I'll go ahead and click on the RNA-seq evidence. And now the page has refreshed and presents you with a list of datasets available in FungiDB. Notice that there are several options of how you can mine this data. And for example, we will take a look at the cells when grown in different media, such as grown in blood. And this particular data set can be mined using differential expression or fold change or percentile. So differential expression, for example, is available for RNA-seq datasets that have at least two replicates, so that we can deploy DSeq to pipeline. However, when samples do not have replicates or the replicates have been pulled, you will notice that this option is no longer available, and you will have fold change query or percentile change query that essentially ranks genes based on their expression. So let's go ahead and run a fold change query on this data set and see how we can set up our search and visualize it. So first of all, now I am on the page that essentially asks you to select your criteria or parameters of your search. I will go ahead and look for upregulated genes. And you know what, I am interested probably in genes that have at least fourfold upregulation. And for my control samples, I will use cells that were grown in liquid media, liquid control media. And then for my comparison sample, I'll choose cells that were grown in blood for 180 minutes. And notice that you have a graphical representation on the right that shows you how your parameters affect your search. And when you click on Get Answer, we expect to return a search strategy at the moment with a single step, something that looks already familiar, similar to how we generate results for ergosterol. And at this step, you can expand your strategy to generate a more in-depth in silico analysis. And there are several options. You can combine this search with other genes, transform it to other records. And notice that when you transform this into other records, there's the shape of the box changes, or you can collocate your results with, say, SNPs and clinical isolates. For our example, let's go ahead and transform these records into orthologs in other species. For example, I would like to look for orthologs in Candida auris. And the question that I'm asking is, out of the 48 genes that we have identified to be upregulated in blood when Aspergillus cells are grown in blood media, how many of these genes have orthologs in Candida auris? And here's another an example of how you can very easily to ask this question. And this particular search is quite powerful because it allows you to mine data sets from other species, either better studied species or for species that have a greater number of data sets integrated and infer data for your model organism. Okay, so right now we will switch gears a little bit, and I will move on to demoing my workspace features. And before I move on, I just want to check if there are any questions, and looks like we are okay. All right.
There was actually a quick question from a user or attendee about, why aren't there any genomes for cordyceps, for example? And I think the bottom line is, we do focus on medically, relevant pathogens, although we are open to including additional organisms depending on their phylogenetic place and obviously whether they're relevant to the community. So the best thing to do is to communicate with us by clicking on the Contact Us link. Let us know about the organisms that you think should be in FungiDB with links to the genome itself if it's available in GenBank. and then we can start a discussion. It doesn't mean it's something we can load 100%, but obviously if it's something that makes sense for our goals and the mission of FungiDB and serving the community, we obviously will seriously consider loading new genomes from new taxa or families.
Okay, that's great. Thanks, Mar. Okay, so next I'll move on to demoing how you can bring in your own data sets and analyze them in Galaxy. And I will start with a general overview of the Galaxy workspace, and then I will pass it on to Amar, who will explain you how you can use RNA-seq data, analyze them in Galaxy, and then import them into FungiDB in order to take advantage of the processes, tools, and various analysis that we provide. Okay, so if you never used Galaxy, or if you don't have an account and you've never accessed Galaxy before, first you essentially will arrive to the sign-in screen, and you will have to go through a few steps to navigate to Galaxy interface. Once in Galaxy, this is how it looks. For those that have used it before, it's very similar to other instances. And Galaxy has several components. You have a top navigational bar that allows you to switch between the main screen or access different workflows or data that is shared with you. You can also browse and deploy bioinformatics tools on the left. And the central part is the working part, is where we provide you with links to various tutorials and learning resources, but it's also the location where you can deploy sample workflows. And the results are displayed here on the left. Normally, if no problems and they have all run successfully, they will be shown in green. So Galaxy is essentially a web-based interface for you to perform a bioinformatic analysis, especially if you don't have experience of controlling bioinformatic tools via command line. And we do offer several workflows, but as you can imagine, there are many different ways to do the same thing. So these are just sample workflows that you are welcome to modify or create completely new workflows for your own sets of data. And Amar, I will go ahead and transfer the screen to you so you can show how to import data into FungiDB.
Okay, great. So while Eve is making me the presenter, just to point out that if you create an account with FungiDB, that same login will allow you to log into the our Galaxy interface, and so you don't have to create a separate login. And we also have a webinar that was fully dedicated to Galaxy, and we go into some basics of how to build your own workflow in Galaxy. So it's incredibly powerful, and I encourage you to watch that webinar and then obviously send us questions as needed. So I'm just going back to Galaxy. I'm already logged in. I'm looking at a workflow here on the right, and so this is just an RNA sequence workflow where I looked at differential expression between two samples in Aspergillus niger. I actually just last night I went to the sequence read archive, and I looked for Aspergillus experiments, and I found this one experiment which was comparing a delta KEXB knockout gene with the wild type, and to see if there are any changes. There are obviously effects on the survival of Aspergillus under high pH conditions and other conditions. But in this case, I was curious to see, well, are there any genes that are changing in expression between these two samples? So I ran this workflow. I picked one of the workflows that are pre-configured here, but you can obviously make your own. And then once I was done, you have all these results which you can download and play around with. But we have also an option for you to export your transcripts per million counts for each gene back into FungiDB. And so I did that. There's an export tool right here, and I just ran an RNA sequence to ViewPathDB exporter. And I just filled up this page here and clicked on execute, and you get a little item here that says, export to ViewPathDB, and it worked. So now in FungiDB, once the export is done, I just go back to the FungiDB page, And I'm logged in here. And so I go to my workspace, and I'm going to go to my data sets. And this will load up a page of all my data sets that I've worked on. And you'll see here, these are different data sets that I've worked on in the past. I can share these data with other users of the database. So this is a nice collaborative tool. And here's the data set from Aspergillus niger, which I exported. The workflow ran overnight, so I exported this morning. I can go to this data set right here. It shows me the data set in FungiDB. This is private. This is only under my account. I can modify the name and change different things about it. I can share it with other users of the database. So I can just click on share and add your email. If you have an account in FungiDB, you will get access to this data. But the really exciting thing about this is that this data now has been integrated for you to actually start asking questions about the data itself within FungiDB. And so, for example, you'll see here there's an available searches. I'm going to click on RNA sequence search. Once you click on this, you will get this search page, which looks a little bit familiar to what Eve was showing you before. You'll notice here I can select the experiment. In my case, this was the A Niger 2 experiment. And once you do that, my samples show up here. So these are the samples that I ran in the Galaxy workflow. And now I can say, well, I'm going to look for genes that are upregulated. And I want to look for genes that are upregulated in my knockout strains compared to my wild type. And if you're interested in learning more about this, we have a whole webinar on RNA sequence searches in ViewPathDB resources, and you can learn more about that there. So I won't take too much time here about configuring the search, but suffice it to say, you can really change it to your heart's content and find a type of regulation that you're interested in. So I'm looking for genes that are upregulated in my 2 knockout samples compared to my wild type samples right here, and I change it to 3-fold. You can change this to whatever number, it's just an arbitrary number. And now when you click on get answer, I get a search strategy, which will give me the results. And hopefully there are results, and they are. So it tells me there are 146 genes that are upregulated by at least 3-fold in my knockout line compared to my wild type. And now you can look at them here. You'll get a graph that shows you a representation of the transcripts per million. And you can either stop here and decide, well, I'm satisfied with this search and I'm done. I have my 146 genes that are really important. But you can actually carry this further and do additional things with it. In particular, what I would like to show you is the ability to analyze the results. Actually, a couple of things. One is you can click on this Genome View, and this will quickly paint your gene locations on the various scaffolds, or in this case, they've been broken up into chromosomes. And so you can see if there's any subchromosomal localization of your genes. In some cases there is, in many cases there isn't, so it depends on what you're working on. You can also analyze your results by doing various enrichment analyses, including GO enrichment, metabolic pathway enrichment, and a word enrichment. These are statistical enrichments, and we have a whole webinar that is dedicated to these types of enrichments, so I would encourage you to go and watch them. I'm just going to quickly run a GO enrichment. I'm going to pick cellular component, but you can pick whatever part of the GO ontology that you want. And you can run all 3 of them as well. So this takes a few seconds, but ultimately what it will generate, it'll generate a table of GO terms, and it will tell you various statistical points about it, including a corrected statistics. But basically, you can see that there's this ion permease looks like it's probably enriched in my set, and that gives you an idea about if there's anything interesting that you may want to go after in your results. And you can edit this always. You can go back and say, well, maybe my 3-fold was a bit too relaxed. I'm going to change it to 4-fold or 5-fold. And you can do this to the point where you don't get any results, obviously, but I'll change it to 5-fold and revise. And so you don't have to go back to configure the search from the scratch. You should just go in and revise it as you wish. And so you'll get your 61 genes, and you can go back and look at them, and your results are here. This graph also is represented on gene pages. So I think, Eve, at some point you'll go to a gene page, but if you run your own experiment, you'll find in the transcriptomics section, there'll be now a new private section for you with this graph loaded there. So it's a nice way to then look at individual genes of interest and explore them there. This is it, Eve. Unless there are any specific questions for Galaxy, I'll turn it back to you.
All right, thanks, Samar.
And I will make you the presenter. All right, go ahead, Eve.
All right, sounds good. Thank you very much. All right, it seems like we are doing good on time. So before we move on to gene pages, and also I wanted to quickly go over Genome Browser, I would like to show you a strategy that I think is quite powerful, and that combines the analysis of your data sort of with the data integrated in FungiDB, but in a slightly different way. So let's say that you have ran a search analysis using your Galaxy data, and this search strategy that's called Magna-Portha Galaxy. These are the RNA-seq results that I have imported, and I have looked for genes that were upregulated in cells, Magna-Portha rhizia cells that were collected from wheat leaves exhibiting signs of disease. And also, I have run separately a signal peptide search in Magna-Portha rhizia. So technically, you can go back, and add a step and say, well, I want to identify signal peptides genes in these group of 5298 genes. But there's also another way to do it. And imagine that you could add a step. And actually, instead of trying to rerun the step that you already created, you simply click on Add an Existing Strategy. And so my strategy that were developed using custom data analyzed in Galaxy is listed here. So you can click on this strategy, and the search analysis will automatically join these two strategies together for you, returning the results for your search. And once it refreshes, we should get a nice observation. It's actually worked for me just a few moments ago. Good. Let me just refresh it one more time. Maybe it's still loading. Well, we will come back to it maybe after the demos, and I can go back to this page. Okay, so let's move on to gene record pages, and subsequently we will also look at visualizing results in JBrowse. So gene record pages in FungiDB or any other ViewPathDB site are essentially encyclopedia-like pages where you can explore all information we have for this particular gene. And obviously for some genes, we'll have more information. For other genes, there'll be less information, especially if they're hypothetical genes where very little known about them. To navigate to a gene record page, you can simply search for a gene ID using the site search window at the top. So for example, I will go ahead and just rerun my search for a search ID. And here we are. This is a result that I will click on, and I'll be redirected to the page. The page has several components. We have a header that lists the gene ID with its function, also gives a name, such as if most genes will have name listed to them, but sometimes it will be listed as hypothetical, for example, and a little bit more information about the location of these genes and the strain. In addition to that, we have shortcuts at the top that help you navigate the records. And if you look at the menus provided on the left, this is quite a long list of the things that you can actually explore. So if you're interested, let's say in transcriptomics data, you can simply click on the transcriptomics shortcut, and it will take you to the RNA-seq transcription summary table with subsequent graphical description of the transcriptome data on the bottom. This particular graph gives you a very nice snapshot of the transcriptomic changes associated with your gene in different experiments, different samples, etc. So it's a nice view to take a look and see what happens to your gene when cells, for example, are exposed to different environmental conditions or isolated from various backgrounds. And if you scroll down and expand the transcript expression table, that section will populate with graphical representation of essentially every experiment that is summarized in this graph. And you can view not only the particular expression of this gene, but you can also view how its behavior changes within the samples corresponding to each data set. You can click, in addition to that, you can click on the links that will take you to publications for these experiments, and you can further use the menus on the left to help you navigate. For example, you can look at the orthologs and paralogs in other species generated by OrthoMCL output, and you can also view the synteny to see how well the gene is conserved in other species, whether they're pathogenic or closely related model organisms, etc. And if you ever want to navigate back to the top of the page, just simply click on the gray button right here, arrow, and it will bring you back to where you have started. So one thing that I wanted to mention is that in addition to this summary information, you can also navigate to GeoBrowse. For example, if you want to look at where the gene is located and explore gene model a little bit more, and perhaps even activate tracks that are associated with data sets integrated in FungiDB. So let's click on the GeoBrowse genome browser link, and you will be redirected to essentially this gene, and I just pre-run this search before. So notice that there are several components to the GeoBrowse window. You have a general menu at the top. You also have navigational menu, and you have a few tracks that is called Select Tracks tab, that when you click, it actually provides you with a list of all tracks that are available for these species. So I'll go ahead and clear results back to the browser. So we have an extensive overview of GeoBrowse actually in previous webinars. So if you want to learn more and cover this subject in depth, I suggest that you listen to that webinar. But just quickly, you can navigate between genomes directly from the genome option. You can also integrate your own tracks by clicking on the track and open tracker file URL, or you can search for a particular sequence if you would like to find something really quickly within this particular genome. You can click on View, and there are several options that help you to customize the view of your GeoBrowse window. And obviously, there's a help menu if you have questions or would you like to learn more. In addition, there is a share button at the very top, at the very right, and let's say you have created a customized view of GeoBrowse and you would like to share that information and the view and the tracks with your colleagues or with your PI. So all you need to do is simply click on the share button to generate a unique URL that can be essentially sent to the people that you're trying to share this information with. In addition, navigation, there are several navigation options that help you to scroll. You can also use magnify or minimize controls to various degrees, or navigate between different chromosomes, or if genes are not assigned to chromosomes, this will be a list of individual contigs. And you can also search and quickly navigate to different genes within GeoBrowse as well. So let's say I would like actually to look up a gene that is annotated with ergosterol. And so the search will be pre-filled with possible suggestions that if you click on, you will be redirected to that very particular location. And the gene can be highlighted if you want to keep your attention on this particular location. And you can also bring in additional tracks. Notice that by default, we list RNA-seq evidence for introns. And this data is calculated using integrated transcriptomics data in FungiDB. You can also activate other tracks. For example, if you want to look at the transcriptomics data, select transcriptomics, RNA-seq, and then let's go ahead and activate RNA-seq coverage plots for the data sets that we have set up a search strategy for the cells that were grown in blood. So I would like to activate unique tracks only, so I'll go ahead and select unique, and then I would want to look at the control sample, and I would also want to look at the cells that were grown in blood media for 180 minutes. And I'll go ahead to click back to the browser. Notice that view has changed, but you can also activate gene density and other various tracks that help you to essentially to look at the gene and supportive information more closely to figure out the answer to your question.
So Eve, sorry
yeah.
to interrupt. There was one question about, since you're looking directly now at RNA sequence data, and that was regarding how can I figure out the 3' UTR of a gene, and this applies obviously to whether it's a 5' or 3' UTR. We do have another webinar that we had a few weeks ago that specifically looked at RNA sequence data in the genome browser and using both the intron evidence tracks and the coverage plots to not only define the gene model and you know whether they're alternative splice versions of a gene, but also to try and figure out how you can use the data to get an idea of where or how long the 5' and 3' UTRs are. So I would encourage you to do that. But as you can appreciate, it's not necessarily an easy task to do on a global scale. If we can, we will do that, obviously, for the genomes that we work on. But we do provide all the underlying RNA sequence data, or many underlying RNA sequence data sets with the coverage plot, which then you can investigate yourself and try and see if you can delineate where is a gene ending or beginning, and that could help you in defining the UTR.
Yeah, that's a great question, actually. Let me show you a track that I think I still have open. Yeah, here we go. That will be a good demo. So notice that for this organism, we have UTRs annotated, but you know things are not perfect. There could be mistakes in gene models. So you as a researcher have to interrogate the data to figure out if this matches your results. And this is a view for JBrowse where I ran publicly deposited RNA-seq data for histoplasma. And this RNA-seq data is stranded data. And what we are looking at here right now is essentially just one track for a reverse strand compared to the gene model that is shown here, along with introns that are shown in the third track right here. And also what I have done, I have activated syntenic sequences in genes, which is it's a track that allows me to see how well the gene is conserved, not only in other pathogens, such as other histoplasma strains, Coccidioides immitis, Coccidioides pasadasi, and also Paracoccidioides species. We have 3 strains. So when you're actually working with the organism where UTRs are not very well defined, if you have a custom set of data, that would be perfect because you can integrate these tracks into JBrowse, and these tracks can be integrated by going to the track, open track or file URL, and that's essentially will allow you to upload a file from your computer. Or if you have analyzed your data in ViewBatDB Galaxy, all you need to do is transfer the data into FungiDB and then bring the bigwig file, which is the coverage for the RNA-seq, into JBrowse session. And then use your knowledge to interpret these results to either confirm the location and the span for the UTRs, or potentially identify new gene models that you need to be aware of based on your results. Okay, are there any additional questions? I see a raised hand.
There aren't any additional questions. If you clicked on the raise hand, please ask your question in the question panel. This way we don't have to try and unmute people and get them to speak. So there's a question panel, just put your question there. I think we answered all the questions so far, Eve. There was one general question about how you can upload your own list of IDs, and I think that was answered, and everybody should have gotten to answer. But if there is time, obviously you can show that, but not critical.
Oh yeah, that's great. Actually, let's just go ahead and demo it. Remember I mentioned that we have specialized searches, and they're available from the searches menu within the header, and you can also find the same searches in the search panel on the left. So if you have a list of genes, you can just start typing gene IDs, and there's an option or a search that is listed underneath the annotation, curation, and identifiers section. And when you click on the gene IDs, you can essentially copy and paste, or if you have a fairly long list of data sets, you can upload your own file. So I'll just create list manually, and hopefully I get the at least a few hits by just manually changing the gene numbers. But once you click get answer, you will see something very similar, right, to what I have shown you before, where your results would be transformed into a first step within the search strategy. And sure enough, my two entries were unfortunately not very good match. So, but it's still a result. I returned one gene that matched my search, and notice that if you're working with a list of genes, depending on what type of data we have available, you can cross-reference with records such as how far these genes are from known retrotransposons, for example, in coccidioides, to see if you can predict changes in the expression. Or you can analyze results and enrich your data using co-enrichment analysis or metabolic analysis, or add step and essentially create a comprehensive in silico analysis using all data in FungiDB for a particular set of genes. All right, so we have a few minutes. So did I hear somebody talking?
Yeah, I was just going to say, so if it would be helpful, Eve, it might be useful. I could demo, or you can demo, showing some of the orthology searches, which might be quite useful. You know, I can demo it with the RNA-seq search I ran before, or you can demo it with one of your previous searches. But there are two, I think, useful searches that might be good for people to know about. One is the phyletic pattern search, which allows you to define the phyletic pattern of genes that you're interested in. So whether you want it to be only conserved within Ascomycota or present, you know, outside of Ascomycota or other organisms, and also the option to transform by orthology between you know, one organism and another, which is quite useful as well.
Yeah, we have transformed by orthology in the first search set, but if you would like to demo synthetic phyletic profiles, that would be great. Okay,
so you can make me the presenter quickly if you want. And there was another question as we were waiting. Somebody's asking whether the database is available freely to anyone, including small companies. The answer is yes. We are a free resource. We're funded by you know, by grants and contracts, so from NIH and from the Wellcome Trust and other sources as well. And so our goal is to make this data available freely to anybody in the community so they can do research and do work. What we don't do is we don't set up private databases for companies or corporations. We don't do that. Our goal is to make the data available publicly as soon as possible to make it most useful. So,
and if you use, sorry, just a quick mention, if you use our data, you found it useful, you generated search strategies or some meaningful output, mention us in your publications or give a shout out or links to gene IDs, which helps us to learn how the databases are being used, and also help to tell us about use cases.
Yeah, very good point.
And of course, try to credit the original data generators too.
Yep. So the search I was going to demo quickly. So if you remember, before I ran an RNA sequence search with my data that I generated in Galaxy. So this data is not available in, we didn't load it ourselves in FungiDB, but I transferred the gene IDs with their expression values. So I could go in here and ask, first of all, I could go in and do a transform by orthology, which Eve showed you in the beginning. And so the question is, are these 146 genes from Aspergillus niger, are they present in another organism? So I can click on select this transform to related records, select orthologs, and now I can go in and select from my list of organisms, whatever I want. So I can start looking at Aspergillus and say, well, I'm interested in knowing if there are any orthologs of these Niger genes in nidulins, for example. And you would know obviously what is a relevant question to your research topic. But once you run this, it'll tell you how many of those 146 genes had orthologs in nidulins, and it'll show you both the original gene that you put in and also the orthologous gene to your original gene. So it's essentially the homolog. So it tells me that these 146 genes have 392 orthologs in nidulins, and then you can go down and look at them, and you'll see here, this is my gene ID in nidulins, and this is the orthology group right here that it belongs to. And so that's a useful tool. The second thing you can do is you can add a step and define the phyletic profile of your gene set. And so I can just go in here, add a step. I'm going to combine my results with another gene search. And in my gene searches, again, we have them categorized, and it may be very intuitive for you, and you'll quickly go and find the category of search that you're interested in. Or you can just use this filter, and I'm interested in something to do with phylogeny. So I just started typing PH, and immediately there's this phylogenetic profile search. So I'm going to explore this. So this gives you this window here, which I can select on fungi. But now I can define my phyletic pattern based on what I'm interested in. So I'm going to expand this different sections here, and I can say, well, I'm interested in genes that are conserved maybe in a number of Aspergilli. Okay, and again, the results may be 0, but this is the nice thing about these in silico experiments. You can run them and you only wasted a little bit of time, but you haven't wasted any reagents. So I'm looking for genes from my previous search that have orthologs in all of these organisms that I selected. And let's say we do not want it to have orthologs in Canada for some reason. So I'm going to click twice on Canada glabrata. And so now it's going to find me any gene that had orthologs in the Aspergillus species I selected, but no orthologs in Canada glabrata. Again, I don't know if this will return any results, but it will find genes that match this phyletic profile, and then it'll intersect the results with my previous search. So we'll let this run in the background, and Eve, we can field any other questions that came up.
All right. I see that there was a question about whether people can integrate their own data for the RNA-seq that was processed through command line analysis rather than Galaxy. I think at the moment we do not have this feature. I'm not a data developer, but I could definitely bring this up to our team to see if this is something that is feasible.
So I can answer a bit in more detail, Eve. We are working on this feature, and it will be coming available hopefully fairly soon. And that's the ability for you to upload your own table as long as it conforms. So, for example, if you have an output that conforms with the format, so let's say it's TPM, transcript per million, as long as you have the gene ID and a column for the TPM values. We're working on a mechanism for you to upload that outside of Galaxy, so you'll be able to do that hopefully fairly soon. That's something that's on our list.
And would this be also for FPKM data or just the TPM?
So that's a good point, Eve. So we are, if you look in FungiDB right now, most of our data is analyzed and it generates FPKM values. But of course, as these technologies develop and people start using different methods, it's become clear that we need to shift away from using FPKM values and moving to something more robust, like transcripts per million. And that's something we are moving to. So in a few months, once we rebuild FungiDB, which is probably by November or something like that, all the RNA sequence data in FungiDB will be represented in TPM values, and we'll be only probably accepting TPM values as far as I know. Although the upload should not care really, as long as you have gene IDs and values right next to them. And so that will be what will be important.
All right, thanks, Samar. So while it's still loading, I would like to share my screen and just kind of go over, quickly show what was the results of the emerging of the two search strategies. Could you make me a presenter?
Yes. Oh, actually, as we were speaking, just so before we, oh, you're presenter now, I think, so that's fine.
Okay, sorry, I'll be really quick. So guys, I just, girls, I just wanted to basically come back and show the type of strategy that I was trying to deploy, but it took a little bit while to load. This is a first step where I ran a RNA seq analysis in Galaxy and imported it in FungiDB and looked for the genes that were upregulated by 5-fold. And notice if you hover over and then just click on the button edit, you can review your search options. And then I essentially combine the step with a previously run strategy listed at the bottom here. And that is a automatically creating a nested strategy by using the strategy available in your account to generate the result of that only 1,000 genes out of the 5,200 that I have identified in the first place have predicted signal peptide. And just really quickly, we are already at the hour. I wanted to mention that in the chat, I have posted a link to our post-webinar survey. And this is very quick. If you take just a few minutes to fill it out, it's a huge help for us, not only to learn how we did, but also to gather information about what topics you will find useful to be covered in the future within our webinars. Amar, and I'll give it back to you so you can.
Yeah, you don't have to necessarily give it back to me. The bottom line is that it ran the search and now it showed me the genes that met the phylogenetic profile, and I think that's no need to show another image of a strategy since we're at the top of the hour.
Okay, sounds good. I guess we will finish up literally in a few seconds. I just want to bring you back to our webinar page that is available under the help menu, learn how to use ViewPathDB. Check it out. We frequently update this page to give you an information about upcoming webinars. If you have questions, or if you're doing something and you're just doing 100 different steps, probably there's a better way to do it, and we will be happy to help you. So email us by clicking on the Contact Us link, and we will try to respond to you within 24 hours to 48 hours to help you out. All right, so thank you very much for everyone attending today and for those who were helping with questions in the back end. And stay tuned for more information from us. Thanks. Bye.
Bye, everybody.